First of all, you will see a lot of signs about the Angioma Alliance. I'm not trying to do advertising of a competing organization. This is a sister organization. I sit on the, I actually chair the scientific advisory board for the US Angioma Alliance. And I like their motto that says, because brains shouldn't bleed, so that I'm putting that on, and forgive me for advertising that. Okay. So the first disclosures, I am mostly funded by the US federal government, the National Institute of Health, Department of Defense. I also have a lot of funding from private organizations and foundations. As I said, I am an unpaid scientific advisory board chairman to the Angioma Alliance. I'm just concluding around where I sat on the advisory council of our government granting agency. So it gave us a good opportunity to really advocate for this disease during a number of years, and I will show you how this has translated into funding. I engage very actively with a number of pharmaceutical companies that are embracing cavernous angioma as their goal, because they bring a great deal of venture capital, and nothing will happen with therapy around the world if pharma does not take on that therapy. So I've made a very careful decision to engage these organizations in a way that doesn't impact my own research, but yet help bring as much interest and money into developing drugs and devices that will help cavernous angioma. And I will not discuss any drugs or devices except in the context of clinical trials. So this concept of deconstructing a neurosurgical disease really is the idea of a lesion that, you know, maybe a decade ago only surgeons understood. And now we have taken two neurologists, pediatricians, biologists, geneticists, so that we are trying to deconstruct this neurosurgical riddle. And the goals of my talk today will be to review the latest mechanisms and signaling aberrations associated with cavernous angiomas. I want to share with you a concept that has become quite prevalent in the United States, but it actually originated in the UK. It originated in Scotland. It's the concept of cavernous angiomas with symptomatic hemorrhage, and why this lesion can be viewed in some ways as almost a separate disease. And I want to talk about how understanding the genes leads us to biomarkers and potential therapies, and the roads we are taking toward therapeutic development and where we are in that regard. So this is kind of what my life looks like. It's kind of an old beat up tree with a lot of branches, and it involves patients of all walks of life from all parts of the world that come to our place and trust us with their care. It involves teams of researchers, collaborations with folks outside. And we've had to adapt a little bit. This is kind of what it looks these days, a lot of Zooming, a lot of masks, but we still have we adapted our response to the COVID-19 by the philosophy that the door remains open and we continue to work with our patients in whatever way we can and advocate on their behalf and continue the activity on the research front. So as I start, I will start and end my talk with a couple of short poems that I think are very important about the overall concept and philosophy that drives our work. Khalil Gibran is a Lebanese poet, and in 1923 he wrote these lines: "What is to work with love? Weave the cloth with threads drawn from your heart." I know each one of you on this call knows what this means. "Even as your beloved were to wear that cloth." And at the end, he says, "It is to charge all things you fashion with the breath of your own spirit, and to know that all the blessed dead are standing about you and watching. Work is love made visible." This is so important because you cannot put your heart in something if it is not really meant to reflect the love you have for humanity. If we were to look at the cavernous malformation, I will use the word CCM and CA. I know you all know those terms, occasionally cavernoma. It started really about 140 years ago with pathologists, mostly in Germany initially, and then later actually in the UK, looking at the lesion as whether it is a tumor versus a malformation on autopsy specimens. And this is a picture on the left from Walter Dandy, one of the leading American neurosurgeons in 1928, where he was tackling this question. And he took a picture at low magnification through his own microscope and had hand drawings on that picture, showing how the lesion insinuates itself in the brain. And he made some very early speculative thoughts about what this could do to the brain. He didn't particularly encourage taking them out because they really were not well diagnosed during life. They were really only known when you ran into them during a surgical emergency or at autopsy. If you fast forward to 2008, you see a very magnified picture of a surgical specimen using MRI at 13 Tesla. You know, 13 Tesla is really a tool that is used only for advanced research. None of you is going to be sitting in a 13 Tesla instrument. But what that will show you is you can see the lesion almost an identical picture to that drawn by Walter Dandy. And it is a way for us to now use the tools of modern medicine and modern science to really shed light on this lesion. But MRI in some ways in the 1980s and 1990s defined the disease because it allowed us to see the cavernous angiomas during life. And the reason you see them so well on MRI is because of the iron that they are associated with because of the leakage of blood, and iron shows up so well on MRI. So you're able to see the lesion during life. And now what do you do with it? Is it a lesion that you leave alone because it's an incidental finding, or is it a lesion that is going to change somebody's life? And this is a big challenge that we face. The next big epoch occurred really about 20 years ago, after the genes were characterized for the familial type of the disease. And you'll understand when we later say, well, what does that matter to me? I have only one cavernoma in the head. Why are the genes so important? So that's one of the things I will explain today. But understanding the genes that cause the familial disease allowed us to understand how a lesion forms as a result of a gene that has malfunctioned. So knowing the gene and knowing the disease allows us to start dissecting what are the steps that result in a lesion forming. And we learned about the concept of somatic mutations, which means a mutation that causes the lesion itself to form. the concept of immune response in the lesion that is very unique to this disease. The concept of phenotype rescue, we didn't really understand what that means, but what that means is that even if you cannot change the gene, if you change how it works, you can prevent the lesion from forming. So you don't have necessarily to change the gene if you can alter some of the other steps that are caused by a gene malfunction. And then this issue of definition of hemorrhage, it is of profound importance clinically and biologically, because we all can live with a lesion that's doing nothing. But then if that lesion switches on and causes a symptomatic hemorrhage, it can destroy our life. So it's a very essential distinction that has to be taken in consideration, even though every lesion has a little blood in it, but yet there is something that switches a lesion from one that just leaks very slowly and does almost nothing to one that's raging in activity and is disabling us. And I think the last 10 years is really what the story I will share with you a lot today. It's been about hammering one nail after the other that we call mechanisms, which are these little switches that are happening to cause a lesion to form, to drive a lesion to progress. And what are the biomarkers on the imaging that tell us what's going on, and also in the blood, and more recently in the stools, in the fecal matter, that tell us what is going on with the lesion and how this influences the natural course of the lesion. We also, in the last 10 years, tackled something that in the UK is a very, very big, important guiding principle, which is what is the evidence that guides our clinical care, and how can we have evidence-based guidelines rather than either dismissive opinions or over-aggressive opinions. You need to have evidence-guided opinions. And where we are with trial readiness and the one active trial currently that is going on in the US. So one important catalyst along this history has been the first investigators workshop that the Angioma Alliance of the United States hosted in 2005. It's hard to believe that it's already been 15 years, but there was very little funding and very little serious biologic research happening until the handful of investigators, you see them here, got together at Duke University, and it was a small meeting where everyone spoke for about half an hour about the research. But for the first time, I, as a neurosurgeon, met somebody who was working on zebrafish and met a person who was working on endothelial cells. And of course, we knew the geneticists already, but we got together and had drinks together. And this idea that the worm, the fish, the endothelial cells can all tell us something about cavernous angioma was never going to happen if we didn't have this meeting in this ugly hotel in North Carolina. But as a result of it, we actually had a call to action with a committee that decided to petition the National Institutes of Health of the United States to put cavernous angiomas on its research priorities. This is a big deal to try to get attention of those folks that are studying 400 diseases, and it's easy for them to dismiss what they might call a rare or inconsequential disease. So we did a lot of lobbying behind the scenes, and ultimately the National Institutes of Health held what they called a workshop, where they invited various scientists, many that were a couple of years earlier at that first meeting. And this was now a chance to present to the government agency what might be research priorities. They asked me to speak on cavernous angiomas, and the late William Young, I'll tell you a little bit about him in a minute, who was doing the same on the arteriovenous malformation fronts. And the NIH didn't think that cavernous angiomas was big enough to deserve its own workshop, so they lumped it into vascular malformations of the brain, which was fine with us, and there are a lot of commonalities and differences. So we proceeded with this, with writing this paper in the journal Stroke a year after we met, and it outlined research priorities. Now you think, what is this exercise going to do? First of all, it sprouts friendships and collaborations. So this is Bill Young, really who did amazing work with arteriovenous malformations, but speaking with him about the differences between AVMs and CCMs and commonalities, meeting in France, meeting in Rio de Janeiro together, are all important ways of driving our thinking into new and creative ways. And then suddenly Bill disappeared because he suffered terminal cancer and died very, very quickly. But his team in San Francisco, it's like you plant the seeds in the ground and it sprouted amazing scientists. You see here Helen Kim next to me. Helen is really the foremost genetic epidemiologists of vascular malformations of the brain in the United States. And she is now a big collaborator as a result of how you sprout these types of collaborations focused on a disease. The idea of collaboration leads to something bigger, which is synergy. And synergy is when two things yield something bigger than the parts. And then cross-fertilization, which is a scientist starting to think about the problem in a different way because of the concepts and ideas of another scientist. So having the endothelial biologist understands what I see in the operating room, and me understanding what does the fish tell us about the cavernous angioma or the mouse, opens really new ways for us. And there were countless meetings like this in Chicago or at Duke with graduate students. Folks, this is Shangbin Shi, who is now in China. And this is how this field kind of moved toward initially starting to define vascular integrity as an issue, and then maybe even the first therapy and the first mouse model. And in France, we had a lot of collaborations. This is Elisabeth Tournier-Lasserve and her group, but also Jean Fleur and Pierre Labauge, neurologists and neuroradiologists, with whom we discovered the idea that the susceptibility-weighted images, which are the highly sensitive gradient echo images, actually show more lesions in the brain than you see with regular MRI. And then those lesions increase in number with age in the familial cavernous angioma situation. And at all times, you have more lesions seen on the new MRI than on the old conventional MRI, giving you a more accurate idea about the lesion burden and maybe a more sensitive biomarker if we're going to measure how the disease is progressing. Canada started with their amazing work on the worm and knocking down the CCM gene in the earthworm, and studying the effects in that very, very simple organism, which told us a lot. And that work has gone a long way toward stuff I will show you a little bit later on transcriptome analyses, and also on the role of zinc in this disease. But more important than the scientist who does the worm or the scientist who studies the endothelium is actually the community that drives our work and hosting us and reminding us through their faces, as I remember today when I look at you across the Zoom, what our work is really all about. Brazil has done the same thing. Of course, they do it with a lot of flair, and they seem to have a lot of fun doing it. And this is a former fellow of mine who worked with me at Yale University about 25 years ago, and now is the lead Cavernoma neurosurgeon and scientist in Brazil, along with a mother and her daughter, and these are always the driving forces. The result of this was an amazing multiplier effect in the funding we received for research. So I told you that we first met in 2005 at Duke University. In 2008, we had the National Institute of Health workshop. And here you see the millions of dollars that started to be generated in research on cavernous angiomas as a result of this advocacy. And working with the director of the National Institute of Health, with a very advocating program officer, is kind of the lobbying behind the scenes that generates this level of funding. And as a result, the number of publications that have arisen in the field. Of course, the story with the UK is extremely important and was playing out at the same time. This is Ian, who is with us today. He was a big force of organizing one of the earliest symposia in London. And I remember coming over with my son, and we had a lovely 3 days in London. And I addressed you all in a small hotel room in London that had approximately 50 people, both clinicians, scientists, and families. And this started to develop along the same timeline as what was happening on the other side of the Atlantic. And as a result of this, you all know this character who works very, very closely with you. But the idea of working with Rustam Al-Shahi and collaborating and putting our ideas together, one of the first fruits of that transatlantic collaboration was the idea that while every lesion has some bleeding in it, there is such a thing called symptomatic hemorrhage that is a little bit different than just the lesion leaking all along. And what we always knew is that many, many lesions are very benign. This is the percent of symptomatic hemorrhage per year, and it's very, very low for a first bleed, unless the lesion has bled already, and the recurrent intracranial hemorrhage rates are extremely high, particularly if the lesion is in the brainstem or deep in the brain, where after a first bleed, the chance of additional bleeds is as high as 30, 40% per year. And that can be devastating. And the idea that don't worry about the cavernoma would not help this person who died from it. So the idea here that the lesion is not always benign was an important thing to put on the table, because the idea that don't worry about cavernomas did not make sense. All of us involved here, our lives were touched by cavernous angioma. Then we go to a doctor and the doctor tells us, don't worry about it, this is nothing. And there is some disconnect here. Well, the reason there is a disconnect, because a cavernous angioma correctly that has never bled has a very, very low risk of bleeding. However, once it has bled, it has 10 times the odds of rebleeding in the next few years. So this is the work that really belongs to your group. This is what Rustam showed in the Scottish population study. And as a result of that, the idea of symptomatic hemorrhage being a different disease than the dormant disease became a very important concept, both for helping patients and for asking biologic questions. What the hell is going on in the lesion that it turned to be like this instead of like that, while everybody has the same gene? The evidence-based guidelines are an important concept, and in 2017, we put together a long list of scientists from different fields. It included Rustam and included many people from pediatrics, from neurology, from genetics. And we went over, this was done a little bit different than the similar exercise you undertook with your organization. We basically wanted to summarize what we knew, even if it was not at the highest level of evidence, because I realized you guys wrote a guidelines that said, we don't know anything. These are the things we need to research. We decided to say, well, you know, out of 1,270 publications, there might be some things where we have consensus about, like, you know, when should you do a genetic test? Does the genetic test tell you anything? Etc. There were consensus recommendations. Several of them reach what we call level B, which is very important in medicine. In fact, almost everything in neurosurgery we do is level B evidence. We don't have level 1 evidence except for 2 or 3 operations we do. So most of neurosurgery is run by level B evidence. So knowing that you can take a lesion out and help epilepsy, for example, was a level B recommendation. And there were some level C recommendations, which is don't do it. So level C means more harm than good. And there was a lot of consensus about, for example, doing unnecessary cerebral angiograms, removing asymptomatic lesions, things of that kind. So we believe that these guidelines were very, very helpful. And you guys should look them up at angioma.org because they remain very, very relevant. So as a result of these guidelines, a lesion that has caused new onset seizures, for example, could be considered for resection as long as it can be done with respecting the important parts of the brain. So this is a lesion that was resected from the left temporal lobe between the main speech areas. And of course, the connection between these speech areas is right here at the depth of the lesion. So it's very important to do this operation in a very delicate way. But if you remove this lesion, the likelihood of seizure control going forward is greater than 90%, and the likelihood of being off medications for seizures is more than 50%. So this particular patient stopped anticonvulsant medications and is living his life free of the cavernoma and free of epilepsy and free of drugs. So this is an important way we can help individual patients. In the depth of the brainstem, it's a major challenge. So we never operate unless the patient is almost getting disabled, and digging into the depth of the brainstem is a difficult undertaking. Of course, we sometimes have splendid results like what you are seeing with this patient. Absolutely perfect. Even her hearing is preserved. However, for each one of these patients with brainstem pathology, there are many, many patients who are in fact heavily disabled and remain disabled after surgery. Surgery as a last resort can sometimes be done. This is a patient who had slipped into a coma after repeated bleeds from a deep lesions that nobody would touch, and she has familial disease and she is elderly. But we operated on her as a last resort. She has recovered to be able to walk with a walker and is able to enjoy the birthdays of her children and is leading a very reasonable life, well better than she would have been if we didn't do it. But this is not a good outcome. We do not want to get to this point. We want to get to a point where the lesion never becomes like this. It stays like this and like this. So surgery outcome, yes, it can be good, particularly if the lesion is superficial in the brain and so forth, but in deep areas and in brainstem, the burden of surgery remains way too heavy and only is being used in individual cases, with many of them paying a high price, but others averting the worst. So what have we learned about the disease biology? And will this knowledge help us treat cavernous angiomas safer and more effectively? So that's what I want to spend the remaining 20 or 25 minutes of the talk about. And basically, we all know there are 3 genes that can cause the familial form of the disease, with the protein coded by that gene really not forming. So it's a loss of function mutation, premature truncation, where the protein that gene would synthesize just does not form. And the familial cases inherit 1 copy of the gene, so the other copy is what makes you normal, until you lose the 2nd copy in a single cell in the brain, and that then has both copies of the gene gone, and then you form a lesion. So even the sporadic lesions, and I'll show you the papers on this, the sporadic lesions that doesn't have the family mutation is due to a somatic mutation of the same genes in the brain. So instead of being an inherited mutation, it's a fluke, it's a chance event, it's a stochastic event that forms 1 lesion. You usually never have such a chance event again, so you only have 1 lesion in your brain. But if you're familial, you have 1 copy already gone. So if any other thing causes the 2nd copy to go bad, you get lesions. That's why the familial cases get multiple lesions, because already 1 copy is gone. So we also learned that the CCM3 disease, one of those 3 mutations, is different from the others. It's far more aggressive. The patients have a huge burden of lesions by their teenage years. Many of them do not procreate and do not have children, so that's why this disease is so rare, the CCM3. And many of the folks have their first bleed in the first 10 years of life, compared to the other familial cases where the first bleed might occur in the middle ages of life. So this had to be discovered by having a whole number of families travel to 1 center where we could study all of them and be able to do this comparison. If every center is seeing 1 or 2 patients and opining about them, we would have never discovered this. And why is CCM3 more severe has become very interesting debate, because it is more severe in the mouse, it is more severe in the worm, but it also causes a very bad gut disorder to have the mutation of CCM3, and as a result, the gut activity goes on further to drive the disease. So this is an example of how watching these cases allows us to ask important biologic questions. What happens if you lose both copies of the gene? Like, you know, you married your cousin. So if you lose both copies of the gene, you cannot live. So you don't even form a heart. This is how important this disease is. So those of us who have the familial disease and have one copy, thank God we have that one copy, because if that other copy was gone as an embryo, you can't even live, okay? So that is so important, because now we understand that if you lose both copies in the endothelium before the organs are formed, you don't even form at all. So this disease happens because the second mutation occurs later in life, after you form the heart and after you formed your brain. So that was a very, very important discovery. And it led us to then look that the fact that the people who live with the familial disease have one copy that's gone, like you see here, but if both copies are gone in any one cell, that cell becomes a cavernoma. So of course, if you lose it in your whole body, you become a cavernoma and you don't live, you know, so that's kind of how that goes. So we had to show this. We had to show this by taking lesions from people's brains at surgery. They needed the surgery, obviously. And we already knew what their germline mutation was. And we had to show that in the lesion, both copies are gone. So we showed this as far back as 2005. And then later, we showed this in a big study with Duke University, where even the sporadic lesions without the family have the same mutations in the sporadic lesion. They just don't have the blood part. They don't have the germline part. So that's key now, because we know that all the lesions form as a result of the same genes. So if we know how to treat the one lesion, we can treat both forms of the disease. It also allowed us to start forming models in mice. This is a very important thing. So we made the mice get second mutations, second hits, and as a result, they form lesions in their brain, and we can count those lesions, and we can put half the mice on a drug, half the mice without the drug, and try to study the effect of a drug on lesion formation, for example. We also developed a model where we knock both copies of the gene after the animal is born. So we bypass the embryonic lethality. And as a result of that, every multiplying cell now becomes a cavernoma. These animals die at about 10 days of age, but their brain is chock full of cavernomas that form within days, because both copies are gone. But yet their heart is good, because we did this knocking out after they were born. And in these first few days of life, we can form lesions very rapidly. And this particular model allowed us to really study a lot of signaling aberrations, what turns the disease off and on, and so forth. This is a mouse in an MRI machine, a little cute coil on the head of the mouse, and we study the mice with MRI, and they have the same MRI appearance as many of you. So there is a lot in common between the tiny little mammal and us, and we're able to study this disease very effectively. What the study of this model showed us is that the mature lesion has inflammation, particularly B cells, which are cells that form antibodies. They're forming antibodies to something, and we'll tell you a little bit about that later. And plasma cells are the ones, the machines that form antibodies. It's chock full of that. AVMs don't have that. Other intracerebral hemorrhage doesn't have that. This is unique to cavernomas. And that inflammatory response is present in the human lesion and in the mouse. There is a lot of proliferation, so the endothelium is actually proliferating in the lesion. And also there are cells that are trying to eat up the blood, so there is a lot of damage going on here. And iron deposit as a result of hemorrhage. All of these things that we see in the human lesion, we're now able to see in the mouse lesion. But there is something more you can see in the mouse, which is the earliest, tiniest little lesion that we never look at in the human brain, because we have no reason to look at those lesions, take them out or anything. So the earliest lesion that has both copies of the gene gone is a bubble. Is a bubble, just a single bubble. It hasn't bled yet. It has no inflammation yet, but it already has lost both copies of the gene. So we have many of these in our head if we have the familial disease, but only one or two go on to be a full cavernoma. And understanding what drives this lesion to become the other is extremely important. So we developed a technique. This is called the micro-CT. It is really available at only some of the big universities. I'm sure it's available at Oxford, Cambridge, maybe in Edinburgh, but that's about it. These micro-CTs are used to study little fossils of insects and worms, but we adapted the one at the University of Chicago to study mouse brains. And with the mouse brains, we are able to count the lesions and assess the volume of the lesions without even cutting the brain. So we can process literally hundreds of these every few weeks and analyze studies from multiple institutions. So we created the core laboratory that specializes in counting cavernomas and counting how big they are, so that institutions throughout the United States, University of Pennsylvania, Duke, UCSD, are doing experiments, and they send all the brains to one place where we assess the results blindly and help give them the answers. So you will see a lot of these pictures from micro-CT on many, many papers published in America, because we created the center laboratory to really do that. As a result of that, the guy that was studying how the heart forms, whose name is Mark Kahn at University of Pennsylvania, tackled the question of why does this gene that causes the heart not to form later forms cavernomas? And he found a switch on the endothelium of the brain that's called MEKK3-KLF2 switch, that is actually active when you lose the CCM gene even later in life. So the heart may have been spared, but if that gene is lost later in life, the CCM gene is lost later in life in a brain endothelial cell, it activates the central signaling. And this resulted in a paper in Nature, because it is extremely important. It was the first disease whereby the same mechanism that causes a major cardiac development and embryogenesis controls lesion development in the brain later in life. So this was a major scientific discovery, and it included groups of people from all over the country working with Mark Kahn on this. We provided him human specimens, and we did the micro-CTs. Also, this led to reconciling the work on endothelium with this central signaling mechanism. So it turns out that this aberration leads to the activation of something called Rho kinase that we knew when it forms, it literally unzips the blood vessels and made them permeable and does not allow them to form tubes. So instead of forming tubes, they form bubbles when Rho kinase or Rho A activity is increased. So all the genes of CCM, when they are lost, cause activity of Rho kinase to increase, which that endothelium impacted by this becomes leaky and unable to maintain a tube formation. Why is this important? This is important because it's for the first time gives us what we call a therapeutic target, meaning if we inhibit Rho kinase, we may be able to prevent lesion from forming even if we don't know how to fix the genes. So nobody's going to change your genes, not today at least, but if we change what they do, then that's a therapeutic target based on the genetic mechanism. So we tested this using a drug that inhibits ROCK. It's called Fasudil. That drug cannot be given to humans for more than a week or two. It is very toxic, but it is a proof of concept study. We were able to give it orally to mice and let them live for several weeks, during which the mice that received placebo formed the same lesions we're accustomed to, while the mice that were fed Fasudil in their drinking water resulted in very little lesion formation, no inflammation, and no bleeding. So it was a very dramatic effect. For the first time in this disease, understanding the biology led to a drug that prevents lesion from growing big and bleeding. So this is important. Even though you can't give Fasudil to humans, now we have a target that we can work with to develop therapies. So as it turns out, there are other ROCK inhibiting drugs that we already have on the shelves, and one of them are statin medications. A statin medication that is commonly used to lower cholesterol, when it is given at a high dose, like with atorvastatin 80 milligrams a day, this is Lipitor, the drug Lipitor, at the dose given for stroke prevention. At that dose, it has very effective ROCK inhibition effect. It's called a pleiotropic effect. Simvastatin, which is a lower dose, does not reach that. It lowers the cholesterol, but doesn't do as good a job in ROCK inhibition. So when we put the mice through these, we showed that atorvastatin at the human approved dose can mimic the effect of Fasudil. And now we can use a drug off the shelf that thousands of people take and test whether it helps cavernomas, whether it helps decrease bleeding in cavernomas. And I will tell you the story of that trial in a minute. The other thing we discovered is that you have inflammatory activity in the lesion, which I told you about, and this results to a very defined immune response with actual antigens, antibody interaction that gives us a chance to form a vaccine or to prevent that interaction as a way of treating the disease. And in fact, if you give the mice the drug that's given in multiple sclerosis or even for psoriasis or for inflammatory bowel disease, these are all B-cell depletion therapies that are used. You get an injection every 6 months and it depletes 50% of the B cells in the body. It's very well tolerated. People take it for arthritis, for psoriasis, etc., and for multiple sclerosis. That drug, when given to the mice, has a very, very strong effect, indicating that B cells can be a factor in driving the lesion. So this led us to dissecting the lesion itself, taking the lining of the lesions like you see here on the left and pulling it out under the microscope. This is called laser microdissection. Look at that, pluck it out. And then you study the RNA and DNA in the lining of the lesion and compare that to in the human, in the mouse, in the worm. And as a result of that, now we know the groups of genes that are activated when the cavernous malformation is formed. Not just the gene that caused it, but all the other things that went awry. So it's like you have one note going bad, then the whole symphony is screwed up, and now we study what are all the other notes that went bad. And one important group of genes for the mature lesions are inflammatory and immune genes. So we can't run away from this inflammation issue. In fact, when you look at a pro-inflammatory state in our body, like being vitamin D deficient. So if you sit not too much in the sun, like you are a lot in the UK, and you don't take your vitamin D, if your vitamin D is deficient, you have a pro-inflammatory state in your body. And we associated this vitamin D deficiency level below 25, which is the same as the level for marking vitamin D deficiency, results in more aggressive cavernous malformation disease. So it is important that this pro-inflammatory state, in fact, is driving the lesions. And when you have less pro-inflammatory state, you're more resistant to the lesions. So we looked at a whole group of molecules in the plasma of the blood that are affected by inflammation, affected by blood vessel formation and remodeling, and said, can these tell us, like vitamin D, about the aggressivity of the disease? And if we analyze these molecules, can we tell you, based on a smart blood test, whether your lesion is going to be active or not active? How many of you, this is far-fetched? All right, let me show you the results. When we combine these molecules in your plasma, in your blood test, in a smart combined way, we come up with 4 molecules that, when combined together, can predict whether you are going to bleed from your lesion next year, the year after your blood test was taken, with a 90% sensitivity and specificity. So I can look at your blood, analyze those 4 molecules, and tell you with 90% assurance whether your lesion is going to stay put or is going to bleed next year. And this is the difference between the blood test of those that bled in the next year versus the blood test of those that didn't bleed next year. So this was a very important discovery in circulation research. And now this blood test is being optimized at multiple institutions in a big study funded by the National Institutes of Health. Because who among us wouldn't want to have a blood test that tells them if their lesion is okay or not? So this is the idea of biomarkers of symptomatic hemorrhage. And we now can take these molecules, combine them in ways that allow us to say, what is the blood telling us about what's going on in the lesion in the brain? This uses Bayesian statistics. Of course, Bayes is the famous English mathematician of the 19th century who developed this concept of weighing observations based on their likelihood. Let me drive you very quickly through these sophisticated MRIs, which you all have heard about, and you say, why the hell do they do them in America and we don't do them in Britain and all of that? So these sophisticated MRIs have to do with measuring permeability. So they are primarily a research tool. They will work in clinical trials, but they do not help the clinician in a day-to-day analysis. But what they basically measure is how leaky is the brain and how leaky is the lesion. As it turns out, the brain, even when there is no lesion, of people with the familial disease has more leakiness. You have a leaky brain, even though you don't have a lesion. And then the lesion itself leaks a lot. It has a lot more activity. And that lesion, when it bleeds, it like breaks the sieve, you know, increases its permeability and leakage immensely, while the lesions that do not bleed, their permeability stays stable. So that means we can look at a lesion and tell you with very good confidence whether it recently bled or not based on its permeability. And we can monitor that over time. The other MRI scan is the one that quantifies or measures the amount of iron in the lesion. It's called susceptibility weighted imaging, turned into quantitative susceptibility mapping. So the black blotch that you see on MRI now becomes a gray scale or a colored scale that tells you how much ink there is in that black blotch. And it is very accurate. It measures accurately how much iron there is. And it's very accurate, reflecting how much iron there is in a lesion. What this graph shows is a lesion that I took out from the brains of patients, and I checked the amount of iron in that lesion in the test tube, and it was exactly the amount of iron measured on the MRI. So it's very good confidence to tell us how much leakage you're having into your lesion, how much iron there is in it. And this is also extremely tight. So it changed by 6% in the QSM means there has been a bleed in the lesion. So if we measure that, we have a very high sensitivity method of telling whether the lesion is leaking or is staying stable. And if you do this biomarker as a way to do a clinical trial, you need only few patients in the trial to measure the QSM effect. But if you wanted to wait to see whether the patient has a stroke, you need hundreds of patients. So this allows us to develop efficient trials to compare drugs, compare doses, and get the best player into the phase 3 trials on the clinic, but do a lot of trials based on biomarkers for proof of concept, so we don't tie up the whole fields and hundreds of patients in drugs that may be useless. This concept is extremely important, and in fact, it led to taking some drugs off the shelf, atorvastatin and propranolol. Propranolol is being studied in Italy. Atorvastatin is being studied with this new biomarker in the US. Tempol is a new drug, never used in human beings before. It is developed by a pharmaceutical company, and they also will use biomarkers as a way to test it. This is the atorvastatin trial. I explained to you why atorvastatin has a biologic rationale, and it is being done only in Chicago. So people travel from throughout the US to Chicago. We pay for their travel and their stay in the hotel, and they come and they get the biomarker image. Then we give them the drug, which is either placebo or atorvastatin. They don't know what they are taking, and I don't know what they are taking. And then we are looking at whether the QSM increases, which would be bad, because if a statin causes your QSM to increase, that means it's making your lesion more bleedy. But if the statin decreases the QSM change, then it is a benefit. So this is a trial that is halfway through now in the United States. We are also starting a trial readiness initiative across multiple institutions. My group is leading that. And what this is doing is taking those same MRI methods and making sure multiple centers can do them, so that the next trials can be done at several institutions and done much more quickly. So you don't then have to come to Chicago. You can choose to go to a closer institution nearby. But this requires that the methods are clearly the same across institutions. So we build a very big, complex study organization for this. But as a result of that, now we can show that these instruments across institutions are all the same when you repeat the test, when you reproduce the test, and so forth. So it tells you that whether you have your MRI in Scotland or in England or in Wales, you you are getting the same measure of iron if you're in a clinical trial. So we are enrolling in both of those studies very well, and despite the COVID crisis, actually, we are moving on with enrollments with these. To close up my talk, I want to just give you a glimpse of a few futuristic concepts. You heard about the microbiome. This was also a story in mice initially, where mice would quit forming lesions when their gut is clean, and when their gut becomes dirty, they form lesions again. And we dissected this very carefully and traced it to a signal from the bugs in the gut called gram-negative bacteria that produce lipid polysaccharides. And the lipid polysaccharides float through the blood, and there is a switch on the brain endothelium in the lesions, where when lipid polysaccharides ties to this, the lesion is driven to grow and bleed. Without the lipid polysaccharide, you can have a lesion, but it will not be very active, or a lesion might not form at all. So this is a very important concept initially in the mice. And we also showed in Science Translational Medicine that CCM3, which is a very severe disease, has a more severe gut problem because of their heterozygous gene in the gut. And as a result of that, their lesions are more active. Now, this paper just came out 3 days ago in Nature Communications. It's the first study of the microbiome in humans with cavernous angioma. And we showed that in order to form a cavernous angioma in man, you need a very special fecal composition. If you don't have that fecal composition, you do not form the lesion. So non-cavernous angioma patients have some stool formation. The cavernous angioma have different stools that allow them to form. It's obviously not the cavernous angioma causing your stools to be bad. It's the other way around. And we showed that because this microbiome causes this lipid polysaccharide synthesis genes to be active, which we showed in human beings for the first time. So now we have a way of looking at what a normal vessel looks like, a vessel that dilates, a vessel that dilates and bleeds, or even a normal vessel that bleeds, and look at what are the secondary switches here that allow one or the other of these changes to happen. And because of these different switches, we have different groups that are working on whether when you are hypoxic and you sleep with sleep apnea, whether that drives the lesion, whether gram-negative infections in urinary tract infection and prostatitis drive the lesion, etc. So we have a lot of very interesting targets, and these are multiple institutions working together to do this. And the final thing I want to tease you with is you guys may know that some elderly people who get an MRI in their brain, they have bleeds in the brain, and their doctors wonder, do they have cavernous angiomas? But in fact, what they have is something called hemorrhagic microangiopathy, which are little leaks in the aging blood vessels of the brain that look very much like a cavernous angioma. And so we looked at the genes. First of all, we look at the permeability of the brain. And you remember that this showed familial patients with cavernous angioma have increased permeability in their brain. Well, so do the elderly patients without cavernous angioma. They also have leaking in the brain. So there is something about this permeability that happens both in aging and in cavernous angiomas. And in fact, when we look at the genes that go awry in aging versus, this is from the aging brain database, versus the cavernous angioma genes, there is a lot of genes in common, a huge amount of genes in common. More than half of the bad genes in aging are cavernous angioma genes. So this disease of cavernous angioma is also playing out very slowly in our aging brain. And when we look at those genes, they are the very same ones that we found in cavernous. This is the mucin gene, that is the gut gene that drives cavernous angioma. TLR6 is a cousin of the TLR4 gene that drives cavernous angiomas. All of these drive cavernous angiomas, and they are also in the aging brain. So maybe our brains, as we get older, all start to look like a cavernous angioma. And the importance of that is that any biomarkers, blood tests, or therapies for cavernous angioma may be helpful to humanity at large and not just for cavernous angioma disease. The big picture is this. This is our team when we ever can get together again. That's what it looks like. This is when we meet with groups from other institutions, and this is where the whole community of researchers are together. It's a multiplier effect, and I want to end with another couple of lines from Robert Frost, also written in 1923. It says that the woods are lovely, dark, and deep, but I have promises to keep. Miles to go before I sleep, and miles to go before I sleep. I think all of us have miles to go before we sleep, because brains should not bleed. Thank you very much for your attention, and thank you for allowing me to share these thoughts with you today.